All right, welcome to Wave Club. And about once a month, I think it's typically the first Tuesday in the month, organized by Josh Jacobs, me, Bart Ermintrout, Uma Mohan, and Irfan Zabe. Um, Today, our speaker is Nick Steinmetz, University of Washington. He's going to talk about brain-wide topographic coordination of traveling spiral waves. Um, Previous talks you can find on our YouTube channel. I guess you can plug that little thing into your phone. Um, Logistics are typically a 40-minute presentation with 20 minutes for questions. And what you should do is use the Q&A feature in Zoom to type your questions, and then we'll bring you on stage at the end, unmute you, and let you ask your question. Each month we'll have a different speaker, and self-nominations are very welcome. So let me say a little bit about Nick Steinmetz. He got his PhD at Stanford in 2014, postdoc at UCL with Ken Harris and Karen Dini. Key contribution was he helped develop NeuroPixels tech. He's an assistant professor at University of Washington. Got his PhD from Stanford in 2014, supervised by Tieran Moore and Kwabena Bohen, and then did postdoc work with Ken Harris and Matteo Carandini at UCL. There he played a role in the development of NeuroPixels technology, and since 2017, he's the program manager for NeuroPixels Consortium. In 2019, Dr. Steinmetz began his lab at UW and Department of Biological Structure and joined the International Brain Laboratory later that year. Dr. Steinmetz is a next-generation leader at the Allen Institute. His research focuses on distributed computations across brain systems that underlie vision and action. His lab approaches these topics with a combination of large-scale electrophysiology and with NeuroPixels probes, calcium imaging across neocortical areas, and systematic optogenetic manipulations, all in combination with sophisticated behavioral tasks for mice. In parallel, his lab also develops new tools to improve the quality and scale of neural measurements. So without further ado, I'm going to stop sharing and let him share his screen. And take it away, Nick.
Thanks, Bard. Okay, let me get my screen sharing up.
Nick, thanks for coming. This is great.
Thanks, Josh. And thanks also to Irfan and Uma. How's this? You're seeing my screen? And let me get the pointer. Oh, pointer is already working. Can you see the laser pointer?
It all looks perfect. Yeah.
Okay. Yeah, so actually, I'm happy to say we have a new department. It's not biological structure anymore. We have a new department as of a couple months ago here at the University of Washington called Neurobiology and Biophysics. So I think UW is going to be, has always been maybe, but certainly, and will be a great place for doing neuroscience going forward. So if you're interested in getting involved with the UW neuroscience community, definitely let me know. Okay, so I'm going to talk about brain-wide topographic coordination of traveling spiral waves. And this talk will be based on a preprint that is really the work of Juen Ye. He's a postdoc in my lab and is a really, really talented and sort of outstandingly creative and productive and just wonderful scientist. So I'm really proud to be here to present his work to you today. So let's get started. So in nature, many systems have organized dynamics, and the particular patterns that those dynamics take depend on the characteristics of the medium of propagation of the interactions between the elements in the system. So that could be, the forces of gravity in the case of planetary systems, or the Navier-Stokes equations in bodies of water. And the particular patterns of dynamics also depend on structural inhomogeneities or structural features of the dynamic medium. So for instance, this spiral pattern of a river only happens when there's a certain configuration of the shape of the bank of the shore and the speed of the traveling water. And then you can get this eddy current forming. So when we're thinking about brain dynamics, we might have several questions in mind. One is, what are the characteristic features of propagating activity in the brain? Because these will depend on both those equations of interaction, which are not in general known for the brain, and because they'll depend on details of the dynamic medium, these cannot be sort of deduced a priori necessarily, and instead must be observed. Secondly, what inhomogeneities of that dynamic medium exist? So what are the features, let's say at the axonal level, that could account for some of the particular patterns of dynamics that we might observe? And finally, what is the extent of that dynamic medium? Are dynamics global or local? In other words, if you have some structured pattern of activity, is that taking place just within the local region that you're measuring, or is it a feature of a more global or more distributed system, which may inform our understanding of both the mechanisms that give rise to it and the way that it impacts perception and behavior? So these are the questions that I'm going to try to ask about the mouse brain today. And to tell you about the particular, to start to introduce the particular phenomenon I'm going to talk about, let's go all the way back to 1929, when Hans Berger performed some of the very first EEG measurements in humans. And you can see one of these EEG traces here. And at the arrow, the human subject closes their eyes. And you can see that counter to the, I think, prevailing expectations at the time, rather than going silent, the brain activity did the exact opposite and became larger in amplitude, at least according to this measurement. And so this oscillation here is the well-known alpha oscillation in the human occipital cortex. And it really illustrates how the brain is a system that spontaneously generates its own dynamics. And this alpha oscillation has been a major focus of research since then. And one of the salient features about it is that it correlates with perceptual behavior. Here's one of the earliest examples of that finding that I'm aware of. So this goes back at least to the 1950s. Lindsay and Lindsley showed that the phase of ongoing oscillations correlated with the reaction time in a task that they had humans doing. And you can see that sort of illustrated here. I'm not going to go into the details of this study, but suffice it to say that this is just one example in a long line of research that even continues today in work by, for instance, Sabina Kastner's group and John Reynolds' group, and many others, relating the phase of ongoing oscillations to perception and behavior. So to try to understand, what might be the neural basis of this kind of relationship, we study mice, and it turns out that mice have an oscillation that has been proposed to be homologous to the human alpha rhythm. And so this oscillation was first described in Einstein-Gulshani 2017, to my knowledge, and you can see it in a whole cell membrane potential recording, this oscillation in the subthreshold regime here at about 3 to 6 hertz frequency range. And in a more recent paper, you can see not only a replication of that strong oscillation in the membrane potential of V1 neurons, but also showing how that oscillation is coordinated between a nucleus of the thalamus and present also in the spiking patterns. And that's both in cortex and in thalamus. So there are several papers about this oscillation, and in one of them from Senzai and Bujaki, their authors made the proposal that this oscillation may be homologous to the human alpha rhythm. So I'm going to refer to this as the alpha rhythm in this 3 to 6 hertz frequency range in the mouse. I am not going to try to defend that claim any further. I'm sure that it's debatable whether this is really like the alpha rhythm. And so if you don't like that terminology, that's fine. You don't have to accept it, but just know that I'll sort of refer to this particular phenomenon as the alpha rhythm or the alpha frequency range in mice, at least for convenience. So in mice, we can measure this activity in this frequency range in a really powerful way using wide field calcium imaging. So in this technique, we essentially do a preparation in which the entire dorsal surface of the mouse's brain is visible through the skull. So this is transcranial imaging. And we can use transgenic mice, which express GCAMP calcium sensitive fluorescent sensor in all excitatory neurons of the cortex. And just by imaging through the skull, we can pick up changes in the fluorescence corresponding to changes in the local local dynamics. And this is not single cell resolution. The spatial scale here is, sort of 10 millimeters from front to back and 10 millimeters across. And so this is picking up population activity patterns. But what you can think of is it's like a sort of very spatially dense measure that's like the EEG in the sense that it's picking up sort of a bulk activity of the neurons nearby each recorded pixel. So it's spatially dense. It does not have the temporal resolution of EEG. So here we characterized with simultaneous electrophysiological recordings the correlation, or really the coherence between the fluorescence measured at some location in cortex and the multi-unit activity or local field potential at that same position in cortex. And you can see that there's a strong correlation between the two things. So this fluorescence is really reflecting the underlying MUA or local field potential up to about 8 hertz or so, 8 or 10 hertz. So we think with this technique, we can get these really sort of beautifully high SNR, high spatial resolution measurements of the local activity that's reflecting the local multi-unit activity of the local LFP, which are tightly correlated with each other. But it only goes up to about 8 or 10 hertz. So that constrains the frequency ranges we can study in this particular experiment. So we were interested to study this type of frequency range that I characterized again as the alpha oscillation. And we were interested to understand at the outset, where does it come from and where does it go? So we thought to do an analysis do an analysis in which we break the activity down by the phase of that signal, of that frequency range at each moment in time and at each pixel on the brain. And so that analysis looks like this. So here is our image of the brain surface, and each of these colored dots over here corresponds to the location from which one of the black traces in the background was taken. So the black in the background is sort of the raw data of the fluorescence in just some example segment. This is this is not an average across anything. This is just one example data segment. And then in this analysis, we filter the data in the frequency range of interest. Here we're using a 2 to 8 hertz filter, and then compute a Hilbert transform, which gives us both an amplitude, which is not shown here, and a phase, which is shown here for each moment in time of that trace. And so as you can see, the filter trace is overlaid in color. And as you can see, this sort of bluish color indicates whenever that pixel is at a peak of the oscillation, and the reddish color indicates whenever that pixel is at a trough of the oscillation, with colors in between to reflect the other phases. And so we can see immediately that if I look at any one time point, say this dashed line, you get different phases at different locations on the brain. So you're at a peak up here in SSP-M at this time point, but you're off the peak here in SSP upper limb, which is over here, and then finally you're at a trough here in retrosplenial and visual cortex. So there's some kind of progression of phases, and we can see that more clearly if we take the phase values measured at every pixel now, not just these sample 7 pixels, but every pixel across the brain, and plot that as a map. So we call this a phase map. So this same calculation of filtering the data over time and then computing the Hilbert transform and extracting the phase was performed for every single pixel, and then that's plotted as a map. And here's the raw data frame at this particular moment. So that's the fluorescence intensity at this one moment. Of course, the phase depends on the signal at adjacent times as well. And when we plot this phase map, we can see a gradient of colors. Again, that's consistent with the phase being different at these different locations. And that gradient of colors goes from sort of red to blue to green, and then over here back around via sort of yellow back to red. And so the fact that there's a gradient of colors indicates that this is a traveling wave, because that indicates that there's a difference in the location, the timing of the peak of this frequency range at different locations across cortex. So if we expand that out, we can see a series of sequential example frames. So this is, again, is not average across anything. And by the way, I should have said the mouse in this case is awake and head fixed, but not doing any particular behavioral task. So in this example data segment, you can see the progression of neural activity, which goes sort of more and more posterior and then comes back around to the anterior side. And that's reflected in this phase map, which shows a gradient that progresses in color from frame to frame as the wave travels. And this kind of traveling wave in wide field imaging has been shown multiple times before. And by the way, in humans, the alpha rhythm is well known to be a traveling wave. I think actually Josh has published on that, if I'm not mistaken. So, the observation that this is a traveling wave was not surprising to us, and indeed was a sort of a sanity check or a confirmation. But we noticed something quite striking in this particular imaging modality and this particular species and condition, that we commonly observed these spiral-like patterns in these phase maps. So here's a zoom in on an example frame where now we've also, in addition to the phase map in the background, we've also performed a motion estimation, frame-to-frame motion estimation algorithm. This is called the Horn-Schunck algorithm. It comes from computer vision, and it just tells you sort of the flow vectors of the activity, of the phase activity from one frame to the next, and allows us to sort of like more easily visualize the direction of travel of these phases. And you can see that over here on the right hemisphere, there's this quite clear circular arrangement of the phases, mimicked by this circular, reflected in this circular arrangement of these flow vectors. And so if we just try to look at this in motion now, I'll characterize these in more detail, of course, in this talk, but just to sort of show you this phenomenon more clearly, we'll go back to this sort of 3D movie, and on the right now is again the raw activity, but on the left we can see that phase map playing out over time, and you can see this spiral of activity that then breaks up, and in a moment will sort of reform again. And so there's the spiral reforming and then even going back the opposite direction. And so what you can notice is that this spiral is something that persists across multiple frames. It sort of breaks up and reforms frequently. Interestingly, in this movie, you can see that the spirals seem to be typically located around this particular location in cortex, and seem to be present bilaterally, and not, for instance, found in this location in cortex. So before I go into characterizing spirals in more detail, let me just show you one more piece of evidence that helps sanity check that this is not some kind of artifact of this measurement modality. So as Bard mentioned, I've been involved in developing the NeuroPixels technology, and the latest version is NeuroPixels 2.0, which has a multi-shank arrangement. So we can actually insert this probe with its 4 shanks into cortex and sample on each of the 4 shanks a little strip of activity. And so this allows us to, by inserting at this sort of shallow angle, we can sample a 2D space of the cortex electrophysiologically. It's only a small part of the cortex, not nearly the whole cortex that we've been looking at with the wide field calcium imaging. But nevertheless, we can check in wild type mouse that do not express any GCAMP sensors of any kind, we can check whether we can see spiral dynamics like this happening in the electrophysiology from this kind of recording. And so here, each little square represents one of the electrical recording sites out of 384 on this 2.0 probe across the 4 shanks, and these are arranged according to their spatial layout in the brain. And again, we've done the exact same thing with the phase analysis, filtered in 2 Hertz frequency, and then plotted the color according to the phase. And you can see, hopefully, that spiral shape in the color pattern happening there, and then moves off the edge of the probe. So we cannot see the spirals persist for long in this modality because, of course, we're just sampling a tiny region of the brain with this approach. But this gives us confidence that there's not some strange artifact of the calcium imaging that's producing this kind of dynamics. Okay, so we go back to the calcium imaging now, and we sought to characterize this spiral phenomenon that we saw. Do these spirals just happen randomly, or do they happen with particular properties in particular locations? And it turns out they do have particular properties and in particular locations. So we designed a spiral detection algorithm that tells us which frames have a spiral and where it's centered. And then this is essentially a spatial histogram of where we find the centers of these spirals occurring. And you can see that they're not randomly distributed. We don't, for instance, find spirals back here centered in visual cortex frequently. Instead, there's a strong peak in the spiral center locations in S1, primary somatosensory cortex, with secondary peaks in M1 and M2. And we can see that clearly if I sort of take a sort of line plot across this 2D spatial map, and you can see this very clear peak in SSP, primary somatosensory cortex, also called S1, primary motor cortex, and secondary motor cortex. And to characterize another sort of basic property of these spirals, if we look at the angular speed as a function of the spiral radius, or the linear speed as a function of the spiral radius, you can see there's a very unambiguous property here, which is that the angular speed is constant as a function of the radius, while the linear speed is increasing. And this characterizes these spirals really as what might be more directly called rotating waves or circular waves, rather than maybe true spirals, in which so basically the entire activity pattern is sort of rotating at a constant angular speed. Okay, so we were really struck by the fact that there's this highly seemingly non-random distribution of these spiral patterns that we were observing. And we got curious about why it would be the case that you might get spirals here as opposed to here. After all, our first order model of the cerebral cortex is that you have a sort of sheet of interconnected neurons with, at least again as a first order model, a sort of isotropic or uniform connectivity across cortex. And in that case, you wouldn't expect any higher probability of finding a spiral here relative to the probability of finding a spiral over here. So we sought to ask whether there might be some inhomogeneity in that sheet of connectivity across cortex, whether in fact there was some non-randomness that gave rise to the fact that spirals, the property that spirals occur there as opposed to somewhere else. And so we had the idea to look at this open data set that was published just a few years ago, in which individual neurons have their entire axonal morphologies reconstructed. So these neurons are reconstructed one at a time, not all simultaneously, sort of one per mouse, I guess. And the entire axonal tree is then registered to the Allen Common Coordinate Framework, so this the same 3D space that we can register our imaging data to. And therefore we can plot, relative to the cortical areas, we can plot the exact spatial position and location of all of these axonal branches. And we did this for a number of neurons, and we started noticing that many of the neurons had sort of non-symmetric axonal branching, that like this green neuron and this sort of periwinkle neuron, they have a major axis of orientation that seems to give them some bias in their direction. And we decided to characterize this axis of orientation for all of the neurons in the sensory part of cortex, and we saw this pattern. So each neuron's major axis of orientation is plotted. So I'm not showing each axonal tree, but I'm just showing the axis of orientation. And it's colored by its angle. And so you can see that the angles of axonal trees, of sort of the structure of the axonal architecture in different places in cortex, really systematically differs. So over here in retrosplenial cortex, you have a lot of neurons that have their axonal architecture oriented this way. Here in visual cortex, it seems to be more a medial-lateral orientation. Out here in the lateral edges, auditory and lateral parts of somatosensory, you get again these sort of anterior-posterior orientations. And then here in somatosensory cortex, we notice this pretty striking arrangement where many of the neurons are really going around in a circle around this location. And so again, we didn't do any selection here. This is all the neurons in this data set. We just literally computed the PCA and took the first, it's literally the first principal component of the axonal terminal locations of these neurons, and just plotted those vectors, and it comes out to really strikingly, we think, mirror the activity flow angle. So this now is a measurement from the activity, from the spiral activity measured in the wide field calcium imaging. And we think that this is a pretty striking alignment. And we have some statistics that I'm not showing here to back that up rigorously, but suffice it to say that this is a really well above chance matching between the angles of these axons and the activity directions. So we think this is quite a clear link between the axonal architecture in sensory cortex and the patterns of travel of propagating activity waves in sensory cortex. I'll emphasize at this stage that the direction of causality here is a bit unclear. It could be the case that some mechanism via development, for instance, sets up these biased axonal structures, and that gives rise to traveling waves that follow that path. Or it could be the case that some other mechanism is directing the traveling waves in this path, and that because the traveling waves are happening over and over again in this particular circuit, that via plasticity mechanisms drives the axons to have this structure. So we don't know the direction of causality. But we did do some modeling that I'm not going to show in detail, although I'm happy to talk about more afterwards, that shows that if you introduce axonal biases like this into a model of a sheet of cortex, it will result in many parameter ranges in having spirals that become centered at the middle of that circular arrangement of axons. And so it's at least the case that this axonal architecture structured in a circle like this in a model will give rise to the axonal, it will give rise to the spiral properties that we observe, specifically the fact that they're centered in the center of somatosensory cortex. Okay, hopefully that's clear. So what I've told you so far is that we have observed this really, I think, we think striking phenomenon that is a really distinctive spiral traveling wave that is very non-randomly localized in the middle of somatosensory cortex, and which matches a feature of the underlying axonal architecture, and is visible in both calcium imaging and electrophysiology. So we think this is sort of a really clear and distinct biological phenomenon. And what we sought to do next was to ask this third question I raised at the beginning, which is whether these dynamics are localized here in sensory cortex versus distributed more broadly. And we thought to ask this question because we knew that this part of sensory cortex has topographic connectivity to other parts of the brain. So in this image, what we're seeing is the results of tracer injections, anterograde tracer injections from the Allen Institute Connectivity Atlas, injected in these locations in sensory cortex. And the colors over here indicate the locations of different tracer injections. And the colors in these other locations, like here in frontal cortex and here in contralateral cortex, indicate the locations of the axons of neurons in the right sensory cortex arriving in those locations. So specifically, the purple location here, retrosplenial, projects both to this purple location here and the purple location there. The red location here in barocortex projects to contralateral barocortex, but also to this specific spot up in frontal cortex. And so what you can see is that this set of regions where these injections were done has a topographic mapping, a reversed topographic mapping, both to frontal cortex and to contralateral sensory cortex. Moreover, it's not only the case that sensory cortex has topographic connectivity within to other regions within cortex, but also that there's topographic connectivity from cortex to the basal ganglia. So specifically, corticostriatal connectivity is topographic point to point, and corticotectal connectivity, so cortex to the midbrain, is topographic, so point to point. So posterior parts of cortex project to this midline part of colliculus, and anterior parts project to this lateral part of colliculus. And I'm not showing it here, but of course, it's even more well known that corticothalamic connectivity is topographic in the sense of one spot in cortex has a particular spot in thalamus that it's strongly connected with. So if these topographic connections exist from these locations in sensory cortex to all of these other brain regions, is it the case that these traveling waves of activity are also shared between all of those brain regions? And we think this is an interesting and non-trivial question. The reason it's interesting is because if it's true that these traveling waves are shared across all of these regions and, for instance, propagate through the basal ganglia, thalamic, midbrain, loop back to cortex and also propagate to frontal cortex and sensory cortex on both hemispheres. If that's all true, it suggests that understanding the mechanisms that form those traveling waves and also understanding the ways that those traveling waves interact with perception and behavior may not be limited to understanding their properties in sensory cortex. We may need to study how they are more globally propagated to understand both the mechanisms that form them and, again, how they are interacting with perception and behavior. It's non-trivial because it's not the case that every pattern of activity is transmitted from any one spot in the brain to any other spot. I mean, first of all, the number of axons that go from one region to another region, one part of cortex to the striatum, is much smaller than the total number of neurons. And so it may be a subset of the information at any one region that's transmitted to other regions. So it may be the case that these traveling waves that exist in cortex are in the so-called null space of activity relative to some of these subcortical regions like basal ganglia and midbrain. So that would mean that some patterns of activity are transmitted from cortex to these subcortical areas by these topographic connections, but not necessarily the traveling waves themselves. Hopefully that's clear. So we sought to test this. And in cortex, the test was pretty straightforward. So we have the measurements directly from both left and right cortex and from some sensory cortex and from motor cortex. And we simply asked, when it is the case that we detect a spiral in, say, right sensory cortex, do we and there's also a spiral in left sensory cortex, is it the case that those spirals are mirrored in their propagation direction, which would match the mirrored topographic connections between left and right cortex? And indeed, you probably don't even have to have me tell you this result, because you can see that there's a very strong left-right symmetry in all of the phases across the whole cortex. And so, of course, it is true that most of the time when there's a spiral over here, there's say a clockwise spiral over here, there's a counterclockwise spiral going on over here. It's a little less clear, but also true that when there's a spiral both in sensory cortex and in motor cortex, these again have mirrored topographic representation. So a clockwise spiral here is matched by a counterclockwise going spiral here, and that's shown here nearly all the time. That's true. So these traveling waves are not localized just to sensory cortex, but are also shared with frontal cortex. But how about those subcortical regions? So to ask that question, we combined our wide field imaging with the NeuroPixels 2.0 electrophysiological recordings that I showed you before. But this time, rather than doing the electrophysiological recordings in cortex, we're inserting the probe deep into subcortical regions, either striatum, which is the input structure of the basal ganglia, midbrain, or thalamus. And when we do that, we get data that looks like this. So we have some activity traces. This is one pixel, but of course we have many pixels of activity of fluorescence measurements in cortex. And then we have the spiking activity on the probe, which you can see just represented here by an example segment of a spiking raster. And we wanted to ask whether this pattern of spiking activity encodes or represents in some way that traveling waves in cortex, those traveling waves in cortex, and in particular, whether it represents those spiral traveling waves. And so the question is, how do we understand whether those traveling waves, those spiral waves, are encoded in this neural code of these subcortical areas, whether this spike raster has some information about those traveling waves. And to do that, we had the idea that what we could do is fit a prediction of the cortical activity pixel by pixel from the subcortical spiking. So we make this prediction and get a predicted trace. So this gray trace now is made by a linear regression fit. So we do a linear fit on other data. This is held out data on other data from the spiking activity to the cortical wide field imaging. So, again, at the basis, literally what we're taking is this matrix of spiking activity and multiplying it by some fit regression weights that gives us a prediction of what the activity in cortex at some pixel is going to look like. And this pixel is very well predicted. Again, this is held out data. So this pixel is very well predicted, meaning that the information about the pattern of activity at this one location in cortex is all contained within this pattern of spiking information subcortically. So then we can go pixel by pixel and make that prediction. So for every pixel in cortex, we can say, now we're going to predict that pixel, now the next pixel, now the next pixel from the subcortical activity. And then we can do the same analysis on this predicted wide field image as we do on the real wide field image and ask whether the traveling waves that we observe in the prediction from the subcortical data do indeed appear in the original raw data, whether those match. And so here's what that looks like. So here's the raw imaging data from one of these example sessions, just a few example frames. And here's the simultaneously predicted. Again, the model was fit on other data than these data. So this is held out data. So simultaneously predicted data from the pattern of subcortical spiking. And you can see that within this sort of region of motor cortex, we get this strong spiral wave. And simultaneously in the prediction, there's also a strong spiral wave. Let me just say one other way you can think about what this analysis is that we did here is that we have this matrix of spiking activity, neurons by time, and we're doing a rotation of that matrix. So this multiplication by a weights matrix that we fit to the wide field data is like a sort of projection or rotation of this matrix into a space that allows us to visualize the activity of the subcortical spiking in the space that matches the cortical space. So these images here are literally a visualization of that matrix of subcortical spiking activity. This is a reprojection of the matrix of subcortical spiking activity in a space that allows us to see that whether those 2D patterns of spiking activity, of traveling wave activity are present in the subcortical spiking activity. Hopefully that logically makes sense. I'm happy to answer questions about it if it doesn't, because to me, this is sort of the main key point. So if there's one thing that I hope you take home from this talk, I hope it's the match between these two things, because I know the spiral waves and the existence of spiral waves are really striking and interesting. But the thing that I think should really change the way that we think about dynamics in the brain and traveling waves in the brain is the fact that these traveling waves are not restricted to the particular place in sensory cortex where we sort of first identified them. Instead, this traveling wave is a global phenomenon. It's present in sensory cortex, both left and right hemispheres. It's present in frontal cortex. It's present in striatum, thalamus, and midbrain. And here's just some statistics to sort of back that up. So this is really, while this traveling wave is happening in cortex, it's also present in all of these other brain structures. And I don't think that we can really think about understanding the mechanisms that give rise to these traveling waves or the way that those traveling waves impact perception and behavior. I don't think we can really think about understanding those in a complete way by only studying the traveling waves in a single structure of the brain, like in cortex. So that, I hope, is the really main take home you take away from here. So what we're showing here is just the spiral matching probability relative to a control analysis. That's just the probability that these spirals are matching between the prediction and the raw data. And again, that's statistically significant in thalamus, striatum, and midbrain. And the phases match as well above a sort of shuffle control. Okay, so the last thing I'm going to show you, which is new relative to the preprint. So I think no one here will have seen, even if you checked out my preprint, no one here will have seen the next part, is how these traveling spiral waves relate to visual motor behavior. So we trained mice on a behavioral task, which is similar to other behavioral tasks that I've used in previous work, and the Carandini and Harris lab has used in previous work, and also this is similar to the International Brain Labs task. So the mouse is seated, head fixed, and viewing a screen in which stimuli can appear on the left or the right side of the screen. And when the stimuli appear, the mouse should turn a wheel. If the stimulus appears on the left, the mouse should turn the wheel clockwise. And if the stimulus appears on the right, the mouse should turn the wheel counterclockwise. And if the mouse turns the wheel the correct direction, then it will get a reward. And it's making this arbitrary linkage between the sensory stimulus and the motor output that is the task of the mouse in this behavior. And we can, vary the contrast of the stimulus and ask about the reaction time of the subject and ask about the percent correct of the subject. So here is the average activity across trials, across correctly performed trials in this task. And so you can see that before the trial starts, there's, on average, close to zero by definition activity. Shortly after the stimulus comes on the screen, we get activity appearing in visual cortex contralateral to the stimulus. So this is a left stimulus trial, and we have activity here in right visual cortex. From there, it propagates, the activity appears to propagate, let's say. The activity appears next in retrosplenial and maybe parietal cortex, and then into somatosensory cortex. This medial part of somatosensory and motor cortex is the limb somatomotor cortex, and then more to this lateral part of somatomotor cortex, which is the mouth and nose part of somatomotor cortex. And then finally, you can see that whereas after the initial bout of activity in visual cortex had little activity, at the end, there's a sort of reappearance of some activity back in visual cortex. So we can look at that in another way by, again, extracting activity from those same pixels that we had looked at in the very first data slide I showed you. And you can see that, again, this is average activity aligned to task onset. You get first this activity in visual cortex, totally unsurprisingly, then propagating to retrosplenial, limb somatomotor, and then mouth and nose somatomotor, before finally a second bump of activity back in primary visual cortex. So on average, on correct trials, this activity appears to take the form of a traveling wave, and in particular, a spiral traveling wave. So here we're plotting this same phase analysis and flow vector analysis from the plots I had shown you before, but on this average activity across correct trials in this task. And because the activity comes back around to visual cortex after traveling through this sequence of visual cortex, retrosplenial, somatomotor, limb, then mouth and nose, and then back to visual cortex, because that's the case, then you get this, the algorithm, the analysis finds this spiral traveling wave pattern of activity. Much more striking to us was the fact that this pattern of activity was extremely strongly correlated with the performance, the successful performance of the mice in the task. So here I'm showing the rate of spirals or of half spirals, which were, say, more reliably detected in correct, incorrect, and missed trials. And you can see there's some background rate of detecting spirals or these sort of half spirals going along this medial half of cortex. There's a background rate of these spirals that happens in spontaneous activity that, those are essentially the spirals that we've been characterizing in previous slides, but then only on correct trials, just after stimulus onset, there's a strong increase in the rate of these spirals that isn't seen on incorrect or on missed trials. So missed trials, the mice just hold the wheel still and don't respond to the stimulus. Incorrect trials, they do turn the wheel, but the wrong direction. So this is super striking to us because basically it suggests that the occurrence of that spiral pattern is related to the mouse's ability to correctly perform the task. Again, causality, not clear here, but that's the observation. So I'm going to just leave you with one slide about some of the future directions that we're thinking about. So we just recently put on bioRxiv a preprint about this new method we have developed in which we can not only do the calcium imaging that I've been showing you, but simultaneously do optogenetic perturbations at any location in cortex. And so this method required both a new microscope design where we have both the calcium imaging light path, but also a galvo-controlled laser light path where the laser can be directed to any spot in cortex through the same imaging path of the wide field calcium imaging. So you get this spot of laser appearing in any spot in cortex. Of course, we can also combine this with the electrophysiology as we have been doing with the wide field imaging and some of the data I showed you. And this allows us to provide a temporally precise and, as you can see, spatially somewhat imprecise manipulation of cortex that's not seen in control subjects that don't have the opsin expressed. And we can do that then at any location in cortex here. If we point the laser at different spots in sort of anterior or posterior cortex, we can create localized manipulations in those areas. So with this technique, we should be able to disrupt the traveling waves and read out whether they have been successfully disrupted or to what extent they have been successfully disrupted. And we hope to write in traveling waves by providing a patterned set of stimulation that will produce a traveling wave exogenously. And we're even working with a control theory lab here on campus to design closed loop control algorithms that may allow us to design that stimulus in a way that is dependent on the behavioral state of the subject to optimally achieve that traveling wave outcome. So in conclusion, what I showed you is that the alpha rhythm is a traveling wave. Again, that was known before for other species. And in particular, it exhibits in mice, in awake mice, these really prominent spiral patterns. The anatomy predicts the structure of these spiral patterns. Specifically, the axonal architecture within S1 mirrors the structure of these waves. And the topographic connectivity between different brain regions leads to these spirals being coordinated topographically across the brain. That's between S1, M1, between left and right cortex, and between cortex and 3 different subcortical areas that we measured. The conclusion that we make from that is that these traveling wave dynamics are global, not local. So specifically, again, I want to sort of reemphasize if there's something that updates about the way that you think about the brain from this talk, I really hope that it's that you think about the mechanisms that give rise to traveling waves and the impact of those traveling waves on perception and behavior as potentially arising not from one site in cortex, maybe from one site in cortex, but potentially arising from this distributed network of brain regions that is simultaneously reflecting this traveling wave propagating activity. So that to me is really what the impact of this work is. And then finally, I showed you that these spiral waves seem to be strongly correlated with a visual motor behavior, which gives, I think, a hint to, and some sort of strong preliminary data that there may be a real functional purpose of these traveling waves, and we can speculate more about that later. Yeah, okay, my last bullet point here. I guess I've said it enough times, but so the perceptual impacts of spontaneous activity and traveling waves may arise from subcortical sites, not just cortex. With that, let me thank again Joanne. So this project, I can't say enough good things about Joanne. This project is all his work from the sort of creative inspiration for nearly all parts of it, and all of the hard work of collecting all these measurements, doing all these analyses, the beautiful visualizations that you've seen. All of it is Joanne's work. Anna Lee and Dan Berman contributed from the lab, contributed to parts of it, and there was some modeling work by Xander Ladd, Nancy McKenzie, and Matt Bull that I didn't show, but I'm happy to go into some detail. We got these transgenic mice from Tanya Daigle. Thanks to funding, especially from the NSF. I have a career award through them that has funded this project, and thanks to my entire lab for their contributions to the intellectual community that gave rise to this. So thanks for your attention. I'm happy to take any questions.
Really amazing talk, Nick.
Yeah.
Thank you.
And there seem to be a lot of questions. So
should I pull up the chat, or how do you want to do it? Yeah, you're gonna call on people, I guess.
Yeah, we'll call on people
Okay.
and coordinate. So first, let's start with a question by Li Hao Yan. I'll put you up on the stage, yeah.
Everyone has access.
Unmute and ask directly.
Li Hao, I cannot hear you. Yes, we can hear you now.
Good. Thanks for your very inspiring talk. I think there are many good ideas there. In particular, I found the link between this anatomical organization of pyramidal cells and the spiral traveling waves very interesting. So I have been thinking about the spiral traveling waves, and it's been found in constant imaging and in local field potential. But that does not imply that there are actual spiral spiking activities followed by the path of these pyramidal cells. So if that's the case, how would that modify this link between the pyramidal cells and the spiral traveling wave?
Yeah, let me go back quickly. Can I easily go back? I don't have a. Okay, we have to flip back. So let me go back quickly. So at the very beginning, what I hope to convince you of with this plot here is that there's a really strong link between the fluorescence activity that we're measuring here and the underlying multi-unit activity. So the calcium sensor is only expressed in the excitatory neurons via our expression strategy, which involves KMK PTA. And the underlying multi-unit activity is highly correlated with that local fluorescence. So I think it's well known from the literature, actually, that the LFP is really strongly correlated with the multi-unit activity as well. But even if you didn't want to believe that, here we have the direct measurement that the underlying spiking activity is related to the fluorescence we're measuring here. So, and I can tell you, I don't have a figure on it, but I can tell you that this oscillation at this frequency, so like when we see this kind of strong oscillations, this is really reflected in, the vast majority of neurons that are recorded spikes with spiking activity locally. So this is really a phenomenon of the spiking activity, I would say.
Okay, sounds good. Thank
Thanks for
you.
the question. Yeah.
Okay, so we'll move on to a question that's in the chat from John Reynolds. John, are you okay with coming up and asking it? Great.
Yeah. Should I ask the question? I think I was told.
Yeah.
First, Nick, a great talk. I really enjoyed it. And I like the conclusion that the intrinsic traveling waves that you see reflect the underlying anatomy. As I mentioned, we've seen something similar. We just published a paper. I have a link in the text showing evidence that they're actually sculpted by long-range patchy horizontal connections. And so this is really consonant with what we're seeing. But I do have a question about the timescales. So in the marmoset, using Utah arrays and looking at the local field potentials, we can estimate using Lau-Muller's algorithm the speeds of individual waves, and they are in the range of horizontal unmyelinated fiber passage velocities, spiking velocities about 0.2 to 0.6 meters per second. And in your paper, you note that the speeds you see in the mouse are considerably slower than that. And I'm wondering two things. One of them is why? Do you think we're looking at the same waves? Do you think this could be maybe a difference in the speed of action potential velocity in the mouse? There is a little cottage industry looking at how brain size and speed of processing go together. So mouse brains work as fast as human brains, more or less. So maybe it's actually a species difference. And I also wanted to get your thoughts on whether the calcium imaging signal could see the sorts of waves that we see in the marmoset, because they traverse the area MT is about 15 square millimeters, and they traverse MT in about 70 milliseconds. So they're pretty fast in the marmoset, at least. And so I was just curious to know your view of the speed of all this.
Yeah, so that's a great question. And I think that the imaging technique we have here is not fast enough to capture, the frequency of activity that you were looking at there. I think one thing one piece of data that I'll point out, and then I'll give a sort of interpretation of what I think about your question. So Joanne has now done an analysis of the patterns of traveling waves in some other frequency ranges that we can measure, which in this case is slower frequency ranges, and you see a similar distribution of traveling wave patterns. So it's similar distribution of spiral centers is what you're seeing here for even slower frequency ranges, which is logical in the sense that if there's this underlying axonal architecture, then activity at any frequency range might be transmitted along them. And so it seems that this newer analysis that I don't have on the slide sort of supports the idea that just activity at any speed is likely to be transmitted along these same pathways. Why this particular phenomenon is slower, I think, could be a difference between the relatively more synchronized state in which this alpha oscillation happens more strongly versus maybe a more desynchronized state that your monkeys are in when they're performing a active behavioral task. So the idea there might be that in the desynchronized state, because neurons are close to threshold firing, then activity can propagate very quickly and is maybe not limited by membrane potential charging, but is instead mostly just limited by the axonal delays. Whereas in this case, where you're in a sort of more oscillatory synchronized state, you may have neurons that sort of need time to charge up their membrane potentials. They may have slower membrane time constants in the first place, but also be further away from threshold, and so that may result in a sort of slower propagation along axonal pathways. So that's pretty speculative, but that would be how I would maybe make sense of that.
Thank you.
Next we'll have Nick Wilkinson, who has a couple of questions, also related to some timing. But
Nick, I cannot hear you. I see you're unmuted, but I'm not getting your audio.
That's out. You getting that?
Yes, there. Yeah.
Yeah. Move these. Sorry, I'm a bit haloed by this light behind me, but so I have a couple of questions. I'll start with the last one. Might the spiral wave be depolarizing and hence engendering the spiking activity rather than caused/constituted by the spiking activity? Can you tell the difference between those two possibilities from your data?
I don't think we can tell the difference between those possibilities. Yeah, we'd need some kind of causal manipulations.
And then again, I'm not sure whether this is something you've looked at, but might there be multi-scale nested networks of these spiral waves?
Like what scales or what kind of nesting do you have in mind?
So if one looks at a river, for example, one can see a large eddy with smaller eddies within it. And I'm wondering if we might see a similar kind of set of dynamics if we looked at different scales, different spatial scales in this data.
Yeah, one thing I can say from the data to speak to that is, Joanne did this analysis of spirals that are detected that have different radii. And, we can detect spirals that are really tiny, meaning a radius of, say, 200 microns, is, I think maybe is, Joanne can maybe correct me, but something like that, up to a radius of, say, 2 millimeters, which is, most of a radius of 2 millimeters would be, most of this sort of part of sensory cortex here.
Yeah.
And one thing that, one of the key analyses that Joanne did there, which I didn't mention, is that you really need to carefully control for the probability of detecting something that looks like a spiral just from spatially and temporally smooth activity. And we do this sort of shuffle analysis in the Fourier domain to do that. And those smaller spirals were sort of consistent with the rate of sort of spirals you would expect by chance from data with the kind of spatial and temporal smoothness as these data have. And it was only when you got to, again, I think about 1 millimeter radius or 1.5 millimeters radius, maybe Joanne can type in the chat or something if I'm getting that horribly wrong, that the rate of the spirals is well beyond the chance rate. And there it's, many-fold over the chance rate, like 5-fold over the chance rate. So highly unlikely to be arising by chance. But it's only those bigger spirals that we really think are this.
So the smaller ones might just be kind of artifacts, really.
I mean, I think it's hard to tell. Like, it would depend on what your definition of sort of a real spiral versus something that could have happened by chance with spatially temporally smooth data is. So I think here, the fact that we have this axonal architecture linkage, I think, and the fact that it's above the chance rate of what you would expect from this kind of spatial and temporal smoothness, I think gives us strong confidence that these particular spirals are like a sort of non-artifactual biological phenomenon.
Sure. My final question, you mentioned these spirals giving rise to alpha sort of range waves. And I wonder whether at the spiral, the core of a spiral wave tends to kind of jitter unpredictably at the phase collapse, fast and unpredictably. And I wonder whether that is creating maybe a gamma range signal that would be kind of much less periodic, and if you see any evidence.
Interesting. So I think the only thing I can really say about that, so we can't measure gamma range activity with the wide field calcium imaging. The only thing I can really say about that is it definitely is the case that as you saw in the movies that I showed, the spiral center appears visually, at least, to sort of drift around during the spiral. We did a little bit of Joanne did a little bit of analysis, sort of looking at the directions and patterns of those drifts, and we didn't notice anything too particularly illuminating about it. But yeah, they're definitely kind of non-stable in that sense. The center appears to move on the spatial scale of 0.5 millimeter to a millimeter.
Yeah, I mean, there's the separable things like the drift of the core and the jitter of the core are separable.
Yeah, I don't think we characterize the jitter. And I guess
maybe
the
you
jitter you're
don't have the
imagining
resolution for that.
is a faster thing than we're talking about yet. Yeah.
Okay, thank you. That's all my questions. That was a fascinating talk. Thanks.
Thanks. Yeah, one thing I'll just very briefly mention. So a lot of people have done calcium imaging in mice with the same kind of prep. Tim Murphy, I think, was among the first, and Chris Neal and others. So there's a lot of really great data sets that have existed long before this one. What I think is different here is that we use these GCAM7 and then even GCAM8 mice, which does have higher signals to noise ratio and higher temporal resolution than so those indicators just have faster kinetics, and I think that's really related to the fact that we were able to discover this phenomenon. Maybe in the future, especially if there's voltage indicators, for instance, maybe we could get even faster measurements of a similar kind, and that would be really interesting things to look at, related to John's question also.
Awesome, thank you.
Thank you, Nick. So next, I'll pull up Michelle Redenbach. She has a question that's related to our calcium imaging discussion just now.
Hi, really interesting talk. Really enjoyed it. My question is somewhat about the practicalities. Like, even if your dynamics are faster, they're still uneven, right, between excitatory and then the sort of like drops that we'd expect later. And I imagine that would have huge effects on your ability to detect the accurate frequency and sort of temporal dynamics of the waves, as well as how they move and their exact phase delays. And then sort of related to another discussion point, in terms of these, like the causality that has sort of come up a couple times, it's in question. I understand that you intend to test this in the future with perturbations, but it seems to me that just perturbing the waves themselves, if they might be caused by neural activity or they might be influencing neural activity, won't really tell you much, even if it changes behavior. You'd also have to manipulate the underlying network structure in some way to really tease apart whether it's, just a consequence of neural activity.
Yeah, thanks. So the first question was about the calcium indicator, and you said that there's how did you characterize it? Asymmetry? You're thinking about the fact that there's like a fast rise time and a slow decay time in the calcium indicators? Is that what you mean?
Exactly. It seems to me that if you have like a long time at the peak, it's going to be very difficult to actually measure a true oscillation, which should be shifting pretty evenly between peak and trough.
So one of the things that we did in processing the data, actually, we have very little data processing here, but the one really key step that we did do is take a derivative of the calcium calcium signal, and that is a sort of poor person's deconvolution. So it essentially emphasizes those increases but minimizes those decreases. And in previous data, we had seen that doing this derivative operation improved the match to simultaneously recorded spiking activity a lot. And so it essentially tries to just be detecting those upward things. I think a sort of proper deconvolution might solve that even more, but I think we get pretty close with this derivative operation. So I think we're relatively insensitive to these slow decay times of the calcium indicator. The second part was about causality and manipulating the network structure. Actually, I completely agree with you. One of the experiments that we proposed in that NSF grant that I mentioned is to perform same kind of experiments actually that Beatrice Theriaud performed in slices many years ago, where you might actually cut some of the axonal structure physically and try to see how that affects wave propagation. So that kind of experiment is certainly also the kind of thing we're thinking about. These are all difficult experiments, but ongoing work.
Great, thank you.
Thank you. related to what you just said about cutting axons, I'll pull up Rainer Engelken to ask his question.
Yeah, thanks. I put in my question before you talked about optogenetic perturbation. First of all, great talk. I was wondering if you cut the corpus callosum, are the waves in the hemispheres out of sync, or if you cut along the coronal plane, so ear to ear in one hemisphere, is the wave interrupted, or would it be kind of inherited from the other hemisphere?
Yeah, it's a great question. So there's a nice paper from Tim Murphy showing that in which was probably the first report of just how correlated the left and right hemispheres are in this kind of imaging from mice. And in that paper, they had found this transgenic mouse line that has callosal developmental atrophy. So those mice don't develop with a callosum, at least not a proper one or something. There, the left-right synchrony is strongly disrupted. So it suggests that the callosum is critical for the left-right hemisphere synchrony. I think the cut experiment that you propose would be a better answer to that, because who knows what else is going wrong in development of these mice. Obviously, many things about their neural activity might be strange. But the cut experiment is really hard because there's a giant sinus right here above the corpus callosum. People do it. Noel Lee has a paper doing it, and I think it would be a really interesting thing to do with this kind of imaging and see what happens. I suppose as for the ear-to-ear cut, the sort of what I interpret you mean is like somehow along this axis.
Exactly.
That's exactly what we're thinking to do. If we have this axonal structure that goes around in a circle like this, can we cut a sort of a branch of that and see if the waves sort of fail to propagate across that or what? Yeah. So that would be the experiment.
Cool. And one follow-up question. So I'm wondering whether your setup would be suitable to test this like communication through coherence hypothesis, or generally, I think also Lauren Frank suggested, or in the lab from Lauren Jack, one work suggested that if hippocampal, I think in their case, theta oscillations are in phase with gait, then there is like information transmitted, and if they're out of phase, there's not. Like I was wondering whether you could use your setup for tackling questions like that.
Yeah, I mean, I think especially the combination of the wide field imaging with the electrophysiology is absolutely a really powerful tool for sort of measuring both of the things you need to measure, both the population dynamics and the spiking activity, to answer that kind of question. Especially this new version we have with the combination of the optogenetics. So yeah, I think that's absolutely the kind of thing one could address with these methods. We have not.
Cool. Thanks.
Thank you. We have a question from one of our hosts, Irfan.
Yeah, I use the privilege of asking my question whenever. And we have many questions, so I'm sure Nick would receive emails, and if you want to continue the conversation offline. But I asked my question because it's following one of the questions in the chat. It's more about the percentage of these waves, how often you see them. And I think I'm curious how often you see these things, and if it's a high occurrence event, do you think just the topography of these axonal connectivities are the only mechanism that can generate these spirals? Do you think if you have a random connectivity network, do you get the spirals in it, and how would you make sure that these waves that you are seeing are just because of those connectivity morphologies?
Okay, so the answer to how can we see them is literally right here. So the peak rate is about 1.5 spirals per millimeter squared per second. It's a little hard to translate that maybe into like because so a millimeter squared, here's the scale bar, right? So 2 millimeters is there, so a millimeter squared, so 1.5 per millimeter squared per second. The short answer there is like they're definitely not happening in every frame, but if you watch these movies, they're happening frequently in spontaneous activity. So yeah, we think it's a reasonably common occurrence, but it's a minority of frames for sure. Hopefully that gives you a ballpark sense, but this is the quantification. Wait, sorry, I blanked already. What was the second part of the question?
Yeah, the second one was whether you think these. So there might be many mechanisms for generating spirals. One can be just these topographical spirals that just because of the connectivity, this is spiral happening, and there many works from more theoretical works, for example, from Bart, that you can see these kind of spirals even if you don't have these spiral connectivities in a random network. You can get these kind of spirals. Just
Yeah,
one
totally.
thought about it.
100%. Yeah. Yeah, I think the main difference between what a random network would produce versus what we see here is just the spatial distribution. And the spatial distribution being non-random and being related to that axonal architecture is presumably there's presumably a causal relationship there. Again, just to repeat what I said earlier, I don't think we know the direction of that causal relationship, and we do have this modeling showing that if you put this kind of circular bias in the connectivity of a sheet of neurons, the model we did was just like a Kuromoto weakly coupled oscillator model, then you will see in that model, under a wide range of parameters, more spirals centered at the middle of that circular bias of connectivity. So that's a sort of existence proof of the possibility that these spirals arise here because of this circular connectivity. It's not a causal statement. It's just a, yeah.
Also, let us know how many more questions you would take if you want.
I don't have anything on my calendar next, so
okay.
Rock on.
Okay. We'll do a couple more. But let us know if you need to go at any point. We'll bring an immediate follow-up question that was just posted from Duigo. If you want to come up and ask.
Oh, yes. Sorry, my camera is not working. So I had a similar question. So we observe spirals also in EFIS recordings across large areas, different regions of cortex, in epilepsy. So my observation of spirals are they're very small percentage compared to other rich spatiotemporal dynamics of traveling waves. So what is the importance of spirals in the context of traveling waves, all different types of traveling waves you observe in the experiments? So why are they important?
Yeah, thanks, Doug. I guess you were a co-author on this Viventi 2011 paper, is that right?
That's correct. And
Yeah.
there are many other follow-up papers
Yeah.
too. So
Yeah.
even in those papers, they were very small percentage.
Yeah. I mean, first of all, it may be I believe that paper was in cats, right?
Yes.
Yeah, so it may be the case that the axonal architecture and organization here is unique to mice or to rodents. I mean, one thing that's worth pointing out is that, in the human, the sort of homunculus is laid out in a linear strip on cortex. And so you have sort of, a sequential progression of body parts represented in cortex, but it's kind of like along the central sulcus in a linear strip. And in a mouse, that just isn't quite true. It's sort of oriented in this circular pattern. So this is limb, somatosensory cortex, mouth and nose, barrel, and then trunk. And so you get this just kind of like circular orientation of the homunculus, the muscunculus, or whatever you want to call it. And so it may be the case that this axonal architecture is just not the same in other species, and that could give rise to more of this phenomenon than in other species.
We also repeated that in mice, like very similar spirals observed, but with very much less frequent compared to traveling more planar-like waves across the cortex.
Which paper was that? I should check out that.
I'll copy it to the chat.
Was it with imaging like this, or was it with
It was in electrophysiology in mice.
Okay, I think the thing that I would point out is that the spatial range is really critical. I don't know if you were, because if you for instance are only going out to say 3 millimeters lateral, then it's going to look like a planar wave, either going anterior, posterior, or posterior,
that's
anterior.
Correct. Yeah.
So you really need this quite lateral part to be able to see that it is a spiral wave.
Yeah,
and one of the analyses that we've done for the resubmission of this paper, we looked at planar waves to some extent, and one of the interesting things that Joanne observed was that, and again, I think it matters exactly how you define what's a planar wave versus what's a spiral and so forth. But one of the interesting things Joanne observed was that the ratio between the occurrence of planar waves versus the occurrence of spirals was dependent on which mouse line we use. We had 3 mouse lines now, GCamp 7, GCamp 8S, and GCamp 8M. and that's from slow to fast there. And the fastest one, the GCamp8M, they have relatively more occurrence of spirals and relatively less occurrence of planar waves. So it's again sort of suggesting that like the temporal resolution and the signal noise ratio is critical for being able to see these phenomena in enough detail to recognize them as spiral waves. I think probably some more investigation is warranted there. And yeah, comparison with electrophysiology would be really interesting. There you have to also think about the spatial resolution. So I don't know. Yeah.
Yeah, thank you. So I totally agree. It will depend on the animal model and also the spatial temporal resolution of the recordings.
Yeah, thanks for the question. Yeah.
Thank you.
Thank you. I have a kind of related follow-up. You had shown, that some of these subpatterns are mirrored in the motor cortex and basal ganglia and subcortical regions. And this nice relationship with visual behavior. I was wondering if you could just speculate a little bit about what the purpose of these mirrored patterns might be in these other regions.
Yeah, I mean, I guess that's the question of just what's the purpose of traveling waves in general, or these spiral traveling waves specifically. I think the kind of natural speculation, especially on the basis of some of the background literature that I showed at the beginning, that as probably everyone on this call knows, there's ample evidence that the phase of oscillations and the phase of traveling waves correlates with aspects of perception and detectability of stimuli and things. So the natural speculation would be, okay, if you have a traveling wave that's going around all of these different sensory modalities, from whiskers to vision to auditory and other somatosensory, then that's sort of transiently altering the sensitivity of each of those sensory modalities to stimuli. And then in the mouse, with this particular structure again of the sensory cortex being sort of circularly arranged, that amounts to kind of like a radar sweep of increased and then decreased sensitivity of different sensory systems as that traveling wave is going around. So that's literally just putting together, on the one hand, this like very common literature report of the phase of oscillations being related to perceptual detectability, and on the other hand, the fact that we see this traveling wave that goes around these different sensory systems. That would be the natural speculation. And I didn't show the particular plot, but we did find that the phase in the visual behavioral task at the end, we did find that the phase of 2 to 8 hertz oscillation, particularly at low contrast stimuli that are hard to detect, is correlated with success in the task. Again, that's a replication, really, of many decades of people finding very similar things. I see Zach Davis is on this call. His work with John Reynolds is one more recent example. So that is a sort of well-known observation. And I think that could be what's happening here. You sort of get a sequential progression of activity that's involved in serially manipulating the detectability threshold. Speculation. Yeah, okay, so then why is that involved in all these different subcortical regions? I guess that my speculation would be that because they are not independent, they're linked in terms of their processing of sensory information and processing of motor information. I think, in my view, I don't know if this is going to sound totally obvious to half the audience and totally heretical to the other half, but in my view, it's not the case that cortex is doing some processing and then basal ganglia is doing some processing and then my brain is doing some processing. There's a loop and they're all doing processing on the information together. So the fact that these traveling waves are distributed just as many signatures of perception and behavior are distributed is just reflecting that fact that this is a sort of a distributed computation where information is flowing around a loop around these areas and is sort of simultaneously and continuously coordinated.
Gotcha. Thank you. That's great. All right. So there are quite a few questions. So maybe Nick, could you put your email in case anyone wants to follow up?
Absolutely. Thanks so much, everyone, for all the interest. This is really good. There you go. And yeah, you can find my lab website online or something. Also give you contact information. So yeah.
Great, thank you.
Well, thanks again for inviting me, and thanks for all the great questions. It's a lot of fun, and looking forward to hearing from any follow-ups. And appreciate it.
I'll probably be emailing you. It was a good talk. I
Thanks,
enjoyed
Bart.
it.
Thanks. Okay.
Thank you, Nick. Thank you, everyone.
See ya.